Okay, so welcome everybody. My name is Mallar Chakravarty. I'm an associate professor in the Department of Psychiatry at McGill, and I work at the Cerebral Imaging Center over at the Douglas Research Center, which is the main psychiatric hospital and facility associated with McGill. I apologize in advance. You might hear a little background noise today, simply because we have a little bit of work going on in the house, which I had not anticipated which I had not known about until this morning. But so if you hear a little some stuff in the background, that's what's going on. So the work today, what I'm going to be doing today is discussing basically an intro to image analysis and image processing. It's a very high-level overview with a specific emphasis on the types of softwares and strategies that are mainly supported by the work that we do over at the Cerebral Imaging Center. Certainly, this extends past the work that you see us do at the CIC. But I do feel like there's some value in kind of showing you, of all the image analysis streams that are out there, what is the stuff that is best supported kind of over at the Douglas for people who want to do their work here. So everything I'm going to say today applies both to humans and animal MRI. But MRI is one of the really, is interesting for a lot of reasons, but one of the reasons why it's interesting certainly is because it's one of the few modalities you can use equally in humans and in animals. And so a lot of the tools that I'm going to talk about today were initially developed in human MRI research and have been kind of back propagated onto what people are doing in animal research. So I think, for all of us, it's really interesting to talk about the organ of interest here. And of course, for the brain, I used to have an excellent RA in my lab. We used to joke, because of his interest, don't take this the wrong way, that everything below the neck was just weak. What he was really interested in was the stuff above the neck and mostly the brain. And it's a unique organ for a lot of reasons. It's, and if you look at the trade-off between the body mass, so 2% of body mass that it typically makes up in a human body, but accounting for approximately 20% of the body's energy consumption, and the little fluctuations that account for functional activations that allow us to do tasks, to access memories, those only account for less than 5% of the baseline activity in any brain region. So what's going on in the brain? Well, in the average brain is about 1.5 kilograms. It makes up, and it's made up of about 90 billion neurons. And so when we use MRI as a way of accessing brain anatomy, brain structure and function, we're really kind of creating these composite pictures of what's going on under the hood, right? And so, as we'll talk about later on, most of the images that we take of the brain using magnetic resonance imaging are really these, they're elegant, high-resolution images, but they're really just snapshots. And they don't really give you much specificity with respect to the neural classes that are underlying these 16 billion neurons that make up the cerebral cortex, which make up anywhere between 20 to 50% of the neurons in your brain. The 150 trillion synapses, the approximately 10 connections per neuron that exist in the brain, or the 160,000 kilometers of white matter axons that connect the different cells within our brain. There are other different parts of the brain that we'll talk a little bit about today if time permits. But of those, the cerebellum makes up almost the other half of the neurons in the brain, depending on who you ask. And despite its relatively understudied position within brain science and neuroscience, and the subcortical structures once again make up about less than 1% of all the neurons in the brain. So sorry, that's the animation that I should have used. So what are we trying to image when we get into our brain images? So there are things that turn over and where there's plasticity in a certain sense in the scale of minutes, hours, or days, such as an ion channel protein, synaptic receptors, single transduction pathways and synapses, and there's frequent turnover in all of those processes. Whereas if you go into neurons, for example, the turnover in neural classes could take up to decades, right? So many of the neurons that you're born with, or some of them, stay in your brain for your entire life, even though you're able to generate new neurons, whereas synapse formation could be more plastic. So for just the quickest refresher of Jamie Neer's talk from last week, I think it's really important to realize what it is you're actually imaging in a standard structural MRI. And as Jamie mentioned in his talk last week, or 2 weeks ago, I should say, on the fundamentals, Gina Kemp asked everyone, it depends on who you ask, but it is possible that there's a lot of wide range of estimates in the number of neurons. So it could be that count is very under-researched, especially with respect to the cerebellum. But yeah, some people believe that up to 80% of the neurons are in there. I think the real count, it's probably about 50-60%, but it's on par with or more than the cerebral cortex. I think the nature and the repetitive nature and the packing of the neurons in the cerebellum allow for the relative abundance of neurons within that structure, which is why that's what we see. Sorry, looking too sidetracked. But I agree, the cerebellum is a fascinating topic. We still don't know what we have there. And yeah, like brain size, the number of neurons in the brain varies quite a bit, and so much of it depends on what technique you use in order to estimate that, as Colin mentions here. So what are we imaging when we use MRI to image the brain? Really what we're imaging is water. So as Jay mentioned, really you're magnetizing water using the MRI as an incredible receiver of radio frequency signals manipulated by an incredibly large magnet. So if 57% of your body is water, we should all be humbled, and we should all be reminded of the fact that more than 3/4 of your brain is water. So next time you think you're very, very clever or very, very smart, just remember that most of our brains is just the equivalent of a couple of cups of water. But that's the property that we're able to exploit in structural imaging, in that hydrogen atoms, because we have a lot of water in our brain, are abundant throughout the system, and as a result, we can exploit the magnetic properties of these atoms because they have spin, they have mass, and they're positively charged. And so, once again, for those who were at Jamie Neer's talk about 2 weeks ago, he went into that in much more detail. So it's a much worse version of his talk. Basically, I just gave him one slide. So when you take brain images, you have a lot of trade-offs and a lot of questions you should ask yourself at some point, at least, as to why you're choosing the modality that you're interested in. And basically, the choice of modality comes down to a trade-off between what kind of temporal resolution do you want versus what kind of spatial resolution do you want. So temporal resolution being, can I get multiple acquisitions of the brain within a confined amount of time, or conversely, over a number of days, weeks, months, or years. And spatial resolution being, what are amongst the smallest pockets of volume that I can resolve using the imaging technology that I want to use. So for example, fMRI, you have elegant FDG PET or different types of PET modalities, and EEG, you have varying levels of high temporal resolution, going from seconds in the case of fMRI, if you consider that your temporal resolution is aligned with the time it takes to acquire multiple fMRI acquisitions, whereas in EEG, you're down in the millisecond range based on the detector sensitivity. So anatomical MRI, for some people, far less interesting because your temporal resolution is basically constrained by the number of times you can enter the scanner in a given period of time. But I think there's still a lot to learn from that. I think also one of the things that's maybe underappreciated about structural MRI is that it's intensely reliable. So with a lot of the algorithms that we're going to talk about today, the test reliability, meaning you put me in the scanner, you take me out, you put me back in, and you acquire 2 images in my brain at each, one image in my brain at each time. Most softwares will give measurements that are about 95, 96% reproducible on the low end in structural MRI. fMRI and certain EEG to a lesser extent, but fMRI certainly suffers from a lot of these issues with reliability, and something to consider when you're looking at longitudinal designs. So we're going to talk about that today. So what's structural MRI? Well, the acquisition is detailed, reliable, and reproducible. There's a lot of analysis streams and a lot of analysis pipelines that are out there, which are very flexible and multi-featured. And I think, like I said before, the underlying biology is far more dynamic and not very static, and much more dynamic than we had ever imagined. Any questions up to this point? Everyone good? I think one of the other exciting things about structural MRI is just how far we've come in a relatively short period of time. So this on the left is one of the first brain images ever acquired of a human brain, and this was acquired on a 0.1 Tesla magnet or system, handmade in Nottingham, where Peter Mansfield was one of the winners of the Nobel Prize for his discoveries in magnetic resonance imaging. And if you can make out a brain in there, you know, congratulations. But what's, I think, very impressive is that if you fast forward about 20 years, from 1978 to, you know, the early, or even less than 20 years, to the mid 90s, maybe even the early 90s, you're starting to go from images which have no contrast, so you can't differentiate which voxels are white matter, which voxels are gray matter, for example, or even where the CSF sulcal spaces reside, to what you have here in the middle, where you have this really elegant differentiation between gray matter structures on the cerebral cortex. You can make out the cortical ribbon very beautifully. Subcortically, you start seeing the striatum, the thalamus, the pallidum, and you can differentiate that against the surrounding white matter, which is very useful. I'm just gonna go ahead and mute a couple of folks. Sorry, guys. I can hear you in the background. All right, but if you have a question, you know, feel free to unmute yourself and interject at any time. Dynamic SMRI is. It's very early in the talk, but I mean, Gina, I'm not exactly sure what you mean by how dynamic SMRI is. Yeah. I mean, longitudinal imaging. I mean, if we have time, yeah. I mean, nothing on. I mean, if you want details on the scale, I mean, that's a bigger talk. I mean, longitudinal MRI can be its own unique talk. This is really meant to be a primer for just the basics of image processing and image analysis. But if we have time, we will get there. It depends on how many questions you ask. Okay. Sorry. As we move forward from 1995 to now, we have a 7 Tesla system over here at the MNI in 2020. Although the first 7 Tesla systems started being implemented in the mid 2000s. You know, you start seeing some really, really beautiful images like you get here, and you're really looking at sub-millimetric resolution, right? So in the span of, you know, 40 years, give or take, we've really come a long way in a relatively young branch of neuroscience, which I think is very exciting too. Yeah, Sylvia?
If you would start a cohort right now, let's say that you would start kind of a longitudinal study, would you go with a 7T, or would you still stick with the 3T? I know that this is kind of a, you know, big question, but maybe in like few ideas of what are really the difference between the 3T and then the 7T, or where would you see some advantages, for example?
So I think right now where we are, and so this is kind of, I mean, there's a number of different types of studies that suffer from this, but there are very few large and longitudinal cohorts at 7T, and that's for a lot of reasons. So with the increase in signal, you also have an increase in the noise floor. And so it's really, really hard to potentially resolve certain regions of the brain reliably. And places that suffer from high amounts of signal dropout are places that are of high interest to us as imagers, which are often at the interface between, gray matter or the brain, CSF and the skull, right? And those little pockets and rapid transitions between tissues kind of create these little magnetization, dark holes, if you will. Well, not dark holes, but the rapid changes cause artifacts that we're still figuring out how to control and deal with. And so what you end up seeing are, in the temporal lobes, for example, in the medial temporal lobes in particular, which is kind of where MRI almost made its bread and butter. There's tons of susceptibility and signal dropout in those regions. Same thing in like the front or the frontal poles and the frontal occipital cortex. Certainly, those are regions that suffer from that. So you have those things to contend with. Now, where you benefit certainly is in terms of resolution, spatial resolution, temporal resolution. And so if you really want to go into detail on specific structures, you could do that. But I think overall, what the challenge really is that there aren't a lot of 7T centers in the world yet. Still, there's a handful in Canada. The first one was at Robarts in London. We're now, I believe, only the second one for human. And unlike the transition from 1.5T to 3T, we haven't really worked out the software for analysis, right? So if you're interested in the magnetic resonance imaging signals, if you're interested in kind of detailed physiology questions, that is an excellent place to start. If you really want to look at, subcortical structures in great detail, that could be a really good place to start. Where, my colleagues who work in 7T, what they suggest is, have a question that you're interested in that you want to go into detail on, and try looking at that through the lens of the 7T, and then try to apply that to understanding what the signals of the 3T are telling. If you want to kind of get some sense of specificity. Yeah.
And then maybe one last question is about, so that's more for structural. What about if you would go, so I thought that if you would go for resting state, fMRI, for example, then there would be a lot of, again, kind of noise that you would have to remove from that. Would you be scared to do rest?
Yeah, but so I think where people benefit a lot is kind of in, people who are we're trying to push in the limits of the scanner and like laminar fMRI or something like that. I haven't seen a lot of, I don't know the literature as well, but I haven't seen a lot of 7T resting state studies, and that's probably for good reason, right? I think the number of people you need to put through a scanner to do that is high to begin with. And then I think the susceptibility artifacts are even more pronounced.
Same issue right there,
yeah. Yeah, exactly. And they're even exacerbated in a typical like EPI acquisition.
Yeah, thanks.
Anyone else have any questions, comments that they want to ask at this point? Okay. Good. So the analysis is flexible, multi-featured. So the interesting thing about MRI is that, if you look at these pictures that I have above, that I just showed, is you have to somehow create a measurement, right? Inherently, these are images, they're pictures of the brain in 3 dimensions. And the important part about that is that it's made up of basically a stack of cubes, each of which has a specific intensity, which we now want to infer where gray matter is, where white matter is, where the caudate and putamen are, for example, right? But we actually don't know where those things are, right? All you have is a picture. So, the basis of any good scientific inquiry is your ability to create measurements. And so the question is, how do you create a measurement? So, in the analogy I like to use is, maybe like my dad joke analogy, is that, if you did, grade 5 science experiment where you wanted to test the hypothesis that sunshine was good for growing your sunflowers. Well, you'd have a control group that had limited amounts of sunshine, and they were put in some sort of covered box. You would have, the active group, which was receiving copious amounts of sunshine every day, and then you'd watch your flowers grow, and you'd measure the extent to which your flowers grew on a daily basis, on a weekly basis. And so your temporal resolution is then defined by the amount of times you stick a ruler next to the flowers that you have. And then once you've done that, you know, if you're particularly precocious 10-year-old in grade 5, you run some sort of statistical analysis to show that sunshine does in fact help you grow flowers at a better and more rapid rate than not having sunshine. And you get a gold star from your teacher, and life would be good. But our ruler doesn't really exist in structural MR, and you have to infer the measurements from the images that we acquire. And so this means that we need to, for example, take our image from the scanner and pre-process it in different ways. So at some level, whether it's at the voxel level or at some reparameterization of the image, we're able to actually deduce measurements that we can use for statistical analysis. So in the most common case, in the easiest case, you can think of the voxel-based morphometry technique, which still gets rapidly used, quite not rapidly, but significantly used today, and we'll talk a little bit about that in next slides. Deformation-based analysis, here on the bottom left, segmentation, which is the automatic identification of specific subregions within the brain, cortical thickness analysis, which we'll talk about in detail. And you can even use the MRI to reparameterize the brain into a surface where you can look at sulcal folds and sulcal geometry. So most of what we do in MRI, this is maybe a bit of an old slide, is predicated on the fact that once you make your measurement, that what you need to be able to do is amalgamate all your data in a common space. And this is because you want to work and make the assumption that whether it's at the surface level or at the voxel level, that if I point to a region on my brain, I want to be able to point to the homologous region in all the other brains, right? And so we'll talk a little bit about how you get into this common space in the next couple of slides. But you know, the one that is nearest and dearest to our hearts here in Montreal is certainly the MNI space. So the MNI space was proposed, you know, in the early 90s, maybe in the late 80s even, by Alan Evans and his group. Here on the left is one of the earliest versions of MNI space, which were taken from 305 young, healthy adults, and were manually aligned to basically to the same coordinate reference frame. And then you do a voxel-wise intensity-based average is performed voxel by voxel. So you get this kind of blurry image without accounting for any of the real anatomy. All you're really doing is accounting for overall brain size or brain shape in this particular model. To that end, you know, this was very heavily used for a long period of time, up until, you know, even the mid-late 90s. And then once computer technology and nonlinear registration technology became more usable, we were able to generate models such as this, where you start seeing really a lot more definition within, you know, for example, that differentiates the subcortical structures. You see more definition in the sulcal definitions in the cortical surface, within the insula, and the cerebrospinal fluids, for example. And so that's, you know, very close to the common space that we're using today. But there are surface-based representations of this as well for people who are doing, for example, cortical thickness analyses that are also commonly used. So here we go. Why would you need to average multiple images? So the idea is, so the question is from Gina is why would we need to average multiple images to create a common space? And the reason is that you don't want to have a single representation of the underlying anatomy. So for example, for a lot of the tools that we use, we do matching to local pieces of anatomy. So I want to match the hippocampus as best I can, but at the same time, I want to match the ventricles and the cortical regions as best I can, right? So if you use one person as the underlying anatomy, how average is that person? What is an average person? Who is an average person? And why should you select that one person? So the idea is that by selecting multiple individuals and finding ways in which you can create the least biased estimates of the global anatomy while truly representing some version of the average anatomy, you're able to keep both some of the population variability and some of the common features in the population available, so that you don't bias downstream analyses in a way that favor, say, the one underlying template that you want to use. Does that answer? Okay, great. It's a good question. So just importantly, the concept of a common space is not particularly a new one. It's one that's been used in neurosurgery for decades, actually even going back to the 1940s, 1950s, using atlases like the Schaltenbrand-Warren Atlas, and just after that, the Talairach Atlas, which I think made its first appearance in the late 70s. And the idea was that you would have a paper atlas that in the case of Talairach, which was the representative neuroanatomy of a single individual, manually mocked up by surgeons and anatomists to show where the anatomy was, and that was manually matched onto each MRI. So the Talairach Atlas is one of the first ones. You might hear often Talairach space and common space and MNI space used interchangeably, and that's because of this historical perspective. Was the representation of a middle-aged, about 60-something-year-old French female who passed away and donated her brain to science. And these are the manual tracings that happened based on the whole axial or coronal sections that were taken of her brain. And the initial common spaces were basically doing these kind of gross and coarse alignments manually based on a coordinate system that was defined by the anterior commissure and the posterior commissure, otherwise known as the ACPC line. And then you chunk up the brain based on the superior, inferior, lateral, anterior, and posterior extents of the brain, and do piecewise linear transformations that best match the atlas or your image to the brain, right? And so the goal of this was effectively to make the atlas usable in a coordinate reference frame for neurosurgery. Oddly enough, this is very similar to what many surgeons still use today as their coordinate reference frame. So just think about that next time you go in for brain surgery, that your brain is being divvied up into coarse landmarks so that you can match to a print atlas, even though there's other technologies that exist. So we get images of the brain, and the question then is, how do we then match our brains in order to do some sort of meaningful group population level analysis, right? Just from the image processing perspective. And so a lot of this stuff, was studied extensively for decades, and I think for most of us, we have a pretty good handle on what happens and how it happens. But a lot of the tools and a lot of the ideas that I'm about to talk to you now are used extensively in commonly used pipelines, such as FreeSurfer, Civet, which we'll talk a little bit about in a while, the FSL tools and so on. And maybe they live under the hood now, and we don't actually see them in as much detail, but it is worth knowing what it is you're doing to your brains. Trizana has a question. Could registration to an average template be removing variability when making volumetric or surface area measurements? So volumetric, well, you wouldn't do your estimations, you wouldn't do your estimates at the level of the average template, right? So let's say, in the coarsest example, use nonlinear registration to map between an average template that has a manual segmentation of a brain on it, and you back propagate that back to the individual in question that you want to study, right? And in the ideal situation, you want to back propagate that into native space, right? And so you're using the unvariable mapping as your baseline, and then you would do everything based on the native space volume. I think I'm answering your question, but I'm not sure. Trizana? I'm gonna say yes, because I don't see anything else. But nonetheless, yeah, okay, surface area measurements. things like surface area measurements and cortical thickness you typically do in using the native space values, even if you map them into a common space. So you don't alter the values based on the mapping, you use the native space values, and you do a matching based on the sulcal anatomy in those cases. So registration, when you acquire an image in the scanner, basically each scanner has its own arbitrary reference coordinate system. And the idea is that every image you acquire, every image you acquire lives somewhere in that coordinate system. So for example, when our MRI tech colleague or our semi-retired MRI tech colleague wheels you into the scanner and make sure you're comfortable, your level of comfort depends on your size, your placement in the scanner, and then where your brain resides in image space relative to everyone else depends on where that image gets taken inside the MRI bore when you're lying in there. Jamie, do you have a question? Do I see you? No? Okay. Just 1 second here. So once you have all the images that you want to acquire, you acquire a lot, then you create some sort of mapping between each of these images, each of these three-dimensional kind of cubes or blocks that we have towards this common space, like MNI space that's been created for you. There are ways to do this within the population itself, but we're not going to go into that too much today. And there's a lot of ways to describe this. An easiest way to think about this is kind of with this kind of shape matching analogy that I'm going to use here today. So if this is this kind of, clunky brain in native space, and here's this beautiful, smooth, oval brain, elliptical brain in stereotaxic space or MNI space. That's another word you'll hear often interchanged with common space or MNI space, which is stereotaxic space. We need to find a mapping between that image into the stereotaxic space. And there's a lot of ways to do this. One is with a linear transformation that accounts for 6 parameters, which is translations and rotations. So around each of the 3 cardinal axes, X, Y, and Z, a global scaling, a scale which is independent to each of X, Y, and Z to get much more finer grained registration. And then you can then apply that to your shape to get a mapping into the MNI space, which is what this is representing here. The idea with this mapping is to maximize the similarity between the images. And you can use an objective function, what's known as objective function, like cross correlation, where you want to maximize the correlation between the 2 images if they have similar contrast, or reduce the sum of square differences to ensure that there's an optimal matching between the brains that the 2 images that you want to register one to the other. So when you have them, then we can use automated algorithms to look at and identify the measurement then, right? So once you have them in a common space, the idea is that we take our measurement and we use that to do some sort of statistical inferencing. So on the left, you have a normal T1 weighted volume. In the middle, you have that classified into gray matter, white matter, and cerebrospinal fluid. On the right, you have the gray matter map, which is then blurred so that you can approximate some of the assumptions in most statistical tests that you want to use. So, for example, let's say I want to figure out how the brain develops from 5 years old to 15 years old. Well, then you can acquire multiple time points from the same individual. And the question you want to ask then could be, how does this brain change as a function of time? Or if you have 2 groups, you can ask questions like, how do these brains differ based on a pathology, or how does the brain development differ in 2 different groups as a function of time, for example. So in a classical VBM analysis, voxel-based morphometry analysis, what you do is you leverage that idea by taking images from the MRI from the scanner, perform corrections for inhomogeneities as a result of the acquisition itself, do your global linear registration, do classification to get gray matter voxels and white matter voxels. You'd blur those, and then you'd run a general linear model or some sort of statistical test that reflects the work that you do, and you correct for multiple comparisons and you correct for multiple comparisons because you have more observations than you do individuals. So you have a multiple comparisons problem, and then you threshold that map for significance. So that's kind of your most basic pipeline that you'd ever want to do. A lot of what I'm saying here applies to virtually any form of image analysis that you want to work on. So that could be functional MRI, PET analysis, diffusion weighted imaging. And so these are kind of the building blocks on just about everything we do in image analysis. Next week, I think what'll be really interesting is we have 4 talented students from my lab. We'll be talking about different multivariate image analysis strategies. And I think for those of you who are kind of reading the more recent MRI literature, just a quick plug for that, because I think that's going to be a really useful talk for a lot of you guys. And just hopefully to provide some intuition on what's going on. So for Haley Park, she's writing, I would also be interested in knowing if there have been, if you've studied important sources of variability based on some factors that can be taken into account to better identify areas. I'm not exactly sure. So why, like, to what extent could you use an understanding of the underlying variability to better improve, say, our segmentations or our classifications? Is that what you're asking? You can feel free to also talk as well, if you'd like. If the common space could actually use the notions of variability, is that what you're asking? Oh, can you make or I mean, the easiest thing you can actually do, and this is very common, especially in the animal imaging literature and the stuff that we do in my lab in animal imaging, is actually you just make a common space from the data that you have on hand. And so you don't have to worry about using a predefined common space. You just, you create one that actually maps onto your population best. So especially if you have an atypical population, so one that we've worked with recently, for example, behavior variant FTD, where you have very atypical atrophy patterns in the brain, that might be a place where, for example, you might want to consider creating your own template. People have done this in Alzheimer's or Parkinson's disease and have found that it's improved their detectability threshold quite a bit. Does that answer your question, Haley? But yeah, if you have questions outside of these, I think we're going to go for another 10, 12 minutes. But feel free to email me. I'm happy to chat about any of these things as we move on. Okay, so other things that you might want to think about, because so much of everything this pipeline depends on contrast. This paper from Christine Tardif, who's a PI over at the MNI, and she published this as part of her PhD dissertation, where she examined, kind of similar to what Sylvia was asking before, the transition between 1.5 and 3T and its impact on commonly used morphometry and statistical analysis packages, and showed that these commonly used acquisitions all have different contrasts. So if you look at you differentiate at this kind of superior edge of each of these images, what you see is very different contrast between gray matter and white matter, for example, and that is going to play out very differently when you actually set out your algorithms to identify where the gray matter ribbon, the cortical ribbon, actually is in the brain. And that's very important for a lot of the work that we do, and specifically if you're interested in detecting group differences. So it's just something to keep in mind, and maybe something you want to keep an eye on when you're designing your own study, for example, for your dissertation. The other thing you might have heard a lot about is the idea of nonlinear registration. That is the idea of normalizing images, so you take away some of the underlying neuroanatomical variability. So, for example, we're going to talk in terms of a source image and a target image, and the idea is to map our source image to look as much like our target image as possible. And so in this particular case, instead of looking at global differences, you're going to look at ways to push, pull, stretch, compress different parts of the brain, so you can get the best matching possible between images. So you can imagine a case where you're looking at a neurodegenerative cohort where you want to expand the ventricles or decrease the size of the ventricles to improve the mapping between two images or your image and your common space. And you apply that deformation field to your source images to get something that looks a lot more like your target image, right? So it's basically an image matching algorithm, but at the level of individual voxel neighborhoods. So just to give a little more intuition, I just pulled this off the internet today, and this is based on one of the most commonly used registration algorithms available. So let's say you want to map the faces of this guy here on the left to this guy here on the right. You fulfill one of the requirements for nonlinear registration, which is you have homology in the anatomy of these two individuals. So they both have noses, they have lips, they have eyebrows, and they have eyes, and they have ears, right? But the shape of their head is very different. The shape of the individual anatomical features are very different. So you have on the left a very kind of square jaw. On the right, you have a very pointy jaw. And the idea would be to map let's say map the image C to best fit image A. And if you do that, you get image C's face looking a lot more like image A's face by compressing the jaw, widening it. And you can do an actually very good job of creating a matched version of that. That's what we're trying to do with nonlinear registration. Same thing goes if you apply the inverse transform from image A, so that you have image A looking a lot more like image C, as you can see here. Or you can pass these through an intermediary image B in order to and concatenate transforms so that you can maybe improve the type of registration that you do, which is a strategy we use in the MagicBrain pipeline in my lab. That deformation field also contains a lot of very valuable information, where the compression and expansion that it takes to map one image to the next actually contains information about volumetric data. And so you'll hear a lot of talk about Jacobian determinants in a lot of imaging literature, and that is effectively the amount of local compression or expansion that had to occur in order to map one image to another image. And that value, that quantity, is a quantitative value that you can use in statistical analysis at the local level. It's often used to modulate the voxel-based morphometry studies that I talked about before. Cortical thickness is another thing that gets used a lot in the image analysis literature. And this is an old example, but a good one, from one of Jason Lurch's kind of early papers, where he identified how you could use cortical thickness estimates to look at Alzheimer's disease populations. And here, again, you rely on that gray white matter classification. Incidentally, the gray white matter classification that lives under the hood in the CIVIT pipeline is a very early implementation of artificial neural network, and it works still very well. You then want to fit a deformable model onto the cortical surface to identify where the sulcal folds are and to identify how you can estimate cortical thickness. So the way an older version of this algorithm works, I don't have an updated slide for it, but it's all very similar in concept, is that basically you have the white matter surface, you have the gray matter surface, and you fit that deformable model to map onto both the white matter surface and the outer edge or the peel surface of the cortex. And when you've done that, you have a white matter surface, and that's basically a surface that is represented by about 80,000 points in space that have a location and a connectivity profile to each of its neighbors. Then you have your peel surface or gray matter surface. And then each of the points on each of these surfaces has a neighbor from which you can measure with your, with your digital ruler what the distance is between the white matter and the homologous gray matter coordinate in the peel surface. And by doing so, you can get a cortical thickness map that looks like this, where red means higher cortical thickness and blue means lower cortical thickness. And once you've done that, you can blur that a little bit so you can you can subject it to statistical testing later on. So here's just an example of how the deformable model algorithm works. You start with an ellipsoid that's defined by a number of triangles connected to each other, which are defined by points in space, and they inherently are flexible by definition, and you're asking them to anneal to the white matter surface of the cortex. And the way that works is that you kind of ask them to do that annealing process over a number of iterations, and when you meet a stopping criteria, you add more points and hence more flexibility to the topology of that surface until you get this kind of crumply, more refined alignment to the white matter surface that you see here until you meet a stopping criteria. And so once you have your white matter surface, you grow that out to look like the gray matter surface. And once you have that, then you have your cortical thickness map. So one of the nice properties of cortical thickness maps is that instead of, one of the things we always do before we run our stats, and in some cases it's controversial, in some cases it's not, I'm happy to talk about this later on too, is you need to apply some sort of smoothing. So in Civit, I believe the default is a 20 millimeter smoothing kernel, which basically just means a weighted average based on some sort of coordinate system. So in volumetric studies, so for example, in a VBM study or in a lot of fMRI studies, for example, where you're not using surface-based models, you'll use this kind of a volumetric blur, which basically means the weighting of the average drops off as a function of a Gaussian distribution, or a Gaussian kernel, I should say, and you weight the stuff at one point higher than you weight the stuff that's in the region. The problem that you have is that by using a 3D coordinate system for something that has a surface representation, is that you then blend values from, say, the inferior parts of the parietal lobe with the superior parts of the temporal lobe, for example. Whereas in many cortical thickness analyses, what you end up using is this diffusion-based smoothing kernel, whereas instead of measuring in a three-dimensional coordinate reference frame, what you're actually doing is you're smoothing in by measuring along the cortical surfaces, right? But Colin, so is there a difference between cortical thickness and volumetric analysis? Okay, there's a lot of questions here I missed. I'm gonna go over them. When you say cortical thickness, is it the same as saying cortical gray matter thickness? It's not, it's a representation of the cortical gray matter thickness, correct? Based on what you see in your MRI data. Ariel Crestel asks, is there a difference in cortical thickness and volumetric analysis? Absolutely, volumetric analysis, the volumes, at least at the cortical level, are considered to be the composite of cortical thickness and local surface area. And so basically cortical thickness is one dimension of the cortical volume. Cortical thickness is thought to be more adaptive to environmental factors and disease processes, whereas surface area tends to be more genetically determined, by processes that happen during, almost the formation of the fetus, really, and even potentially in the early developmental period. And Colin says, all the cool kids are now doing their fMRI surface smoothing on the surface. We learned from the free surfer type folks. Thanks, Colin. I don't know that literature as well, but I think that from what I've seen, that's actually true. A lot of people are doing surface-based smoothing for that type of work. I think I'll just make one little plug for segmentation. And so the idea for segmentation is kind of automated identification of structures in the brain. I think this is all we'll have the time for today. Things tend to spread out quite a bit when you do these kind of things on Zoom. But basically, at a fundamental level, and there are much better algorithms for this now, but at a fundamental level, segmentation is just the propagation of labels manually generated on some sort of high resolution atlas, like the ones I was talking about before, but they could be other ones. And it's the process of using nonlinear registration to match anatomies between two MRI scans and then propagate that label from the ideal representation to the subject that you would like to analyze using the instructions given to you by the nonlinear registration. I have a whole bunch of other slides, but we have 5 minutes left and I don't want to go late. I think more advanced topics will be covered next week by, like I said, a number of really bright students from the lab. But if you guys have any other questions, I'd be more than happy to answer them right now. I have the hand up. Can you feel free to jump in? Nope. Okay. Well, in machine learning techniques? Yes, Saranya? Okay. I'm not sure that the question is, a machine learning techniques that use cortical thickness, surface area long, is what a confound, Saranya? Depends on how many subjects. Okay, so the question is that based on what I mentioned before, where you have volume is basically the product of surface area and cortical thickness, it is using all three a confound in your analysis. And essentially, I mean, I can jump ahead to some of the stuff that I was hoping to cover today, but yeah, there's redundancy. So what ends up happening is that you don't really get extra information from the cortical thickness. Is it a confound? No, because your machine learning algorithm should be able to do some sort of feature selection to pick maybe better features, but it depends on the sample size and the method that you're using. Katie's asking me, can you talk about the difference between segmentation and parcellation? So for the fMRI folks, which I guess is the question you're asking, segmentation really is having the idea of this anatomical underlying prior that you want to represent after the fact. The idea of parcellation, and there's a lot of ways to define parcellation, and that can be its own 1 hour talk, the debate between segmentation and a proper way to do parcellation. But parcellation can be anatomically driven. So segmentations are a subset of parcellations. And so, you know, where you're trying to recapitulate some version of the gyral anatomy, the functional anatomy, the myeloarchitectonics, whatever. But you can also do it atheoretically, right? So dropping 1,000 equally spaced or equally sized parcels on the brain is also a parcellation, right? So in the ideal situation, anything you find in one of your connectivity analyses would be reproducible across parcellation schemes. I'm not sure if that answers your questions. Oh boy, could you please cite, oh, that's for citations now. Which type of softwares are common for registration, segmentation, and cortical thickness using MRI, please? I'm thinking about using FreeSurfer, but there might be others. So FreeSurfer obviously is very common. The Civet tool out of the MNI and Alan Evans group is very common. That's what we use in my lab. ANTS has a very elegant cortical thickness tool. I guess the research standard for nonlinear registration and segmentation is mostly the ANTS tool package right now, although there are certainly other very good tools out there. I can provide some literature on this, and there are specific advantages and disadvantages to using different packages, which you should be aware of as well, and including their susceptible, like where you're susceptible to false positives and false negatives, and where they're susceptible to artifacts as well. But again, you know, we could go on for this. The goal of this was to provide a bit of intuition on what's out there and what's going on beneath the hood when you do some of your image analysis work. All right, it's 12. Any other questions? Or if anyone wants to interject, ask a question, raise their hand. Now would be the time. Thank you. If not, I thank you all for your attention. Enjoy the rest of your day. Thanks for coming, and we'll be back next week. See you later.